Our last abstract for the early afternoon session will be presented by Dr. Gregory Baker. The title of his talk is Mechanisms of Innate Immune Escape Mediated by Glioma-derived Galectin-1. So we're welcoming Dr. Baker back again this year after he gave a great presentation last year. But he will be talking about his work with glioma, and particularly looking at glioma cells overexpressing Galectin-1, and how that this seems to have an effect on suppressing NK cell immune surveillance. And I believe today is going to be talking more about how they may be actually working indirectly through its anti-inflammatory effects on monocytic cells.
Okay, thank you very much for that kind introduction. Before I begin, I'd just like to thank the Symposium Committee for allowing me to speak here again today. I'm really excited to be presenting to you some form of an update on something I presented here at the first annual Immuno-Hematology Symposium last year, where we have identified a novel mechanism by which malignant brain cancers can suppress the innate immune response. So this is a bit of a juxtaposition to some of the other talks that we just heard about the adaptive immunity and how tumors can suppress the adaptive immune function. We'll switch gears and we'll talk about innate immune function right now. So I have no conflicts of interest to disclose. Okay. So last summer we published a paper in the journal Cancer Research where we were able to show that natural killer cells were able to effectively eradicate cancers of both rat and mice origin if we were to make those cancers deficient in a carbohydrate binding protein called Galectin-1. So this is a summary slide of a lot of the data that was published in that report, and if I can begin with just looking at panel A here. Panel A is a Western blot of whole cell lysate of a commonly used malignant cancer cell line that we use in mice. It's called GL26. It's a malignant glioma. And we fluorescently modified this cancer cell line to express M-citrine fluorescent protein so that you can visualize these cells using fluorescent-based microscopy. And what we can show here is that using Galectin-1 specific hairpins, we can knock down Galectin-1 expression by roughly 60% in these cancer cells, compared to a control cell line, which we refer to as non-target, which is a scramble shRNA control, and also compared to the parent cell line. Now, if you look at B is telling you that there's no deficiency in these cancer cells to proliferate like normal if you grow them in a tissue culture cabinet. So Galectin-1 doesn't appear to suppress the ability of the cancer cells to proliferate under normal conditions in vitro. However, what we found very early on is when you engraft these cancer cells into an immunodeficient mouse model called RAG1 knockout, which lacks adaptive immune function but still has innate immunity, what you find is that all of these cancer cells that are Gallin deficient fail to cause lethality in this mouse model. Even past 100 days post-engraftment, there's no evidence that these cancers are lethal. And if you do the histology after this 100-day time point, what you find is no evidence for tumor outgrowth whatsoever. And this is very much opposed to the two control cell lines, which invariably cause lethality around day 20 post-engraftment. So at that time, we speculated that it was the innate immune response that was perhaps responsible for rejecting these cancers. So to test that hypothesis, we obtained NOD-SCID IL-2 receptor gamma null mice, or NSG for short. These are one of the most immune-compromised mouse strains that you can purchase at this time. We get them from the Jackson Laboratory. And not only do they lack adaptive immune function, but they also lack innate immunity. So what happens is when you engraft Gal-1 deficient glioma cells into the brain of this mouse strain, you find that those cancers grow like normal. They cause lethality within day 20. And this is very similar to what you would get if you put a non-target control or a Gal-1 expressing cancer in. And again, this is juxtaposed to the RAG1 knockout model where Gal-1 deficient cancers are rejected and the ones that express Galectin-1 grow out. So if we move back into the RAG1 knockout model, again, and graft galectin-1 deficient glioma cells into the brains of that mouse, and then treat those mice with immunodepletion antibodies specific to natural killer cells, what you find is that all of those mice that receive the immunodepleting antibody against NK cells all grow normally Gal1 deficient glioma cells. And compared to all the controls that were treated with rabbit anti-serum, all of those cancers are again rejected. So not only did this data suggest that we have a strong innate immune component to the rejection of these cancers, but it appeared to be natural killer cell specific. So also at that time, we were able to show that Gal1 deficient glioma cells are also somehow sensitized to NK-mediated tumor lysis. And we could show this in vitro in a few different ways. One of the first things that we did was to use DIC imaging on the confocal microscope. And this is a 22-hour time-lapse movie of a 50-50 mixed ratio of Gal1 deficient cancer cells that have been labeled with a green dye, shown here in green, as opposed to the cells that, let's see if I can point to a few of them. These cells here that are unlabeled with the green dye, these are cancer cells that express galectin-1. Now if we hopefully get this movie to play. Okay, or not. No problem. Anyways, what you would find is that many of the green cells begin to become lysed by many of these natural killer cells. These little dots that you see here, these are all natural killer cells, which was an early indication that somehow the Gal1 deficiency was making these cells more of a target to natural killer cell cytotoxicity. And luckily, I also have some concrete data to show you that if you do this in a more quantitative way, you can indeed show that Gal1 deficient cancer cells are much more susceptible to tumor targeting by NK cells compared to those that express galectin-1. So before we stopped and published this work, one of the sort of burning questions that we asked very early on was whether or not we could use recombinant galectin-1, add it to the culture media to see if we can inhibit NK cell mediated attack against these cells. And the answer to that question is no. It's a resounding no. If you add this is quite a high amount of recombinant galectin-1, and we don't see any deficiency in the natural killer cell's ability to target those cells. So that was quite paradoxical at first. And the rest of my talk is going to be showing you mechanistic insight as to that potentially explains why galectin-1 doesn't influence natural killer cells directly. So one of the first questions that we had was whether or not a galectin-1 expressing cancer cell can somehow rescue the cell death phenotype that we see in a Gal1 deficient cancer cell. To test that question, we did mixed ratios of Gal1 expressing cancer cells together with Gal1 deficient cancer cells and increased the ratio of the Gal1 deficient cells from 20% in that co-implant to 50% and all the way up to 80%. So in this red line we have 4,000 cells which express galectin-1 and we have 16,000 cells which do not express galectin-1. And what we found was very striking. As a function of the amount or the percentage of galectin-1 deficient cancer cells in that co-implant, you could dose-dependently increase the survival of RAG1 knockout mice, all the way to the point where if you had 80% of those cancer cells Gal1 deficient, those animals would be cured of the disease. So even past 100 days post-implantation, you'd euthanize these mice, you'd section those brains, and you ask, do I see any Gal1 expressing cancer cells? And the answer was no. So it suggests the following paradigm. Somehow Gal1 deficient glioma cells are pro-inflammatory. They're initiating an NK-mediated response. However, once that NK-mediated response is initiated, there is somehow indiscriminate tumor lysis that we're seeing. Because again, this data showed that there was not even any Gal1 expressing cancer cells in the brain of these mice after day 100. So this suggests, or the conclusion from this experiment, is that galectin-1 somehow a priori blocks the ability of the innate immune response to recognize the cancer. So moving on, we can show actually proof positive that these Gal1 deficient glioma cells are indeed pro-inflammatory. So these are all cytokine arrays, which basically is a Western blot that has 40 different representative cytokines that you can apply either conditioned media from your glioma cells or whole cell lysate. or indeed, in this instance, whole brain tumor homogenate of that microenvironment. And what we can show is that as a Gowin deficiency actually very much increases the amount of CXCL10 or IP10, which is a very interesting cytokine to have raised because it's a potent natural killer cell chemokine. So it basically gets turned on. The non-targets express very little of it, both in both situations here, not only in secreted conditioned media, but also in whole cell lysate, you see IP10. So if you do a very similar experiment, like I mentioned before, in the tumor microenvironment, you start to see things, in addition to IP10, which is confirmatory, we start to see things like MCP1 and MCP5, which are potent myeloid chemoattractants. So we were interested in understanding more about the nature of the types of immune cells that would infiltrate these cancers. So to test this, we would implant non-target or Gowin-expressing cancer cells compared to their Gowin-deficient counterparts. And after so many days, we'd euthanize those mice, digest the brain tumor material, isolate single-cell suspensions, and then analyze them by flow cytometry. And this is a gating strategy, flow cytometric gating strategy, performed 48 hours post-tumor implantation. In step 1, I'm showing you this is whole brain cells. Step 2 and 3 are doublet discrimination gates, so we're analyzing single cells. And step 4 is a CD45 gate. So this is a pan-leukocyte marker. So this little bump that you see high upstream, it represents the immune cells that are infiltrating that brain tumor microenvironment after 48 hours. If you take those CD45 positive cells and then further stratify them based on 2 sort of prototypical myeloid cell markers, one's called GR1, the other being CD11B, you can stratify many different types of cells using that gating strategy. And what I can show you through the quantitation here is that we get roughly 2.5 times more total infiltrating leukocytes into a Gowin-deficient tumor microenvironment compared to a tumor that expresses galectin-1. And what's most striking, as a subset of these total immune cells, if you look here, this GR1 high, CD11B high cells, or this sort of density that you see here, we see 7 times more of that infiltrate in a Gowin-deficient glioma cell compared to a non-target after 48 hours, which was quite striking. But if we consider the natural killer cells, we see a non-statistical difference in the number of those cells that are penetrating the tumor after 48 hours. And if you consider the y-axis, okay, compared to this myeloid population, which has an order of magnitude higher than these NK cells, we get an appreciation for just how many more myeloid cells are penetrating these cancers early on compared to the natural killer cells. But this all changes when you do a very similar analysis 24 hours later. We start to see meaningful increases in the number of natural killer cells. And this graph represents nearly a 10-fold increase of the number of natural killer cells in a Gowin-deficient microenvironment compared to that which is Gowin-expressing. Now, we also included in this analysis anti-granzyme B antibodies. Granzyme B is a cytotoxic granule that's produced by effector lymphocytes, such as natural killer cells and T cells. But I remind you that we're doing this work in RAG1 knockouts that don't have T cells. So we can show very nicely through flow cytometry that these NK cells are indeed expressing granzyme B compared to isotype control, which is shown in the gray silhouette here. And conversely, we see no such increase in granzyme B expression in any of the other myeloid cells that we're analyzing, which is to be expected. So we asked a very pertinent question at the time. We said, what is the role of the GR1 positive myeloid cell in the phenotype that we're seeing? So we're seeing rejection of a Gowin-deficient glioma, but we're seeing this massive recruitment of myeloid cells that sort of predates the recruitment of natural killer cells. So what is the function of the myeloid cell? So to address that question, we immunodepleted GR1 positive cells using specific immunodepletion antibodies in RAG1 knockout mice. And what we were able to show is that after 7 days of treatment with anti-GR1, Gowin-deficient glioma cells would grow much larger compared to their control cohort. As well, we could also show that the granzyme B expression, or coming from the natural killer cells, within that tumor microenvironment is much attenuated compared to the control. So it implicated a very important role for the myeloid component in somehow mediating or sort of somehow helping this natural killer cell immune response. So because we now understood that there might be at least a minimum of two distinct cell types that are mediating this rejection, we decided to take a reductionist approach by FACS purifying out just the cell types that we thought might be implicated in the rejection that we saw in vivo, which was to include circulating GR1 positive myeloid cells, both polymorphonuclear as well as mononuclear, as well as the circulating natural killer cells. And then what we would do is we'd co-culture those cells together with tumor cells that either express galectin-1 or not. And then after so many hours, we'd analyze the activation of those cells using flow cytometry. And this is a summary slide of many weeks' worth of in vitro analysis. Just to suffice to say that what we found from this series of analysis is that when you co-culture monocytic, specifically monocytic, not granulocytic myeloid cells, together with natural killer cells and Gowin-deficient glioma cells, what you can show is the phenotype of those sort of monocytes goes from what I'm showing you here in this orange box of the typical monocyte phenotype on H&E has a very scant cytoplasm, kidney bean nucleus, and after just overnight incubation with these two other cell types, these monocytes in these three examples I'm showing you here become larger, they become highly vacuolated, they begin to have notched nuclei, amorphic nuclei, and begin to start forming cellular processes, which is sort of an indication that these cells are somehow becoming activated. We can further show, using a prototypical dendritic cell marker, CD11C, that these myeloid cells, these monocytic myeloid cells, are expressing far more CD11C, specifically in response to Gowin-deficient glioma cells compared to those that express galectin-1. And it's very easy to show from the literature that dendritic cells are potent activators of natural killer cell effector function. So this is an interesting speculation. Moving forward, there's still a lot more work to be done in this area, but it's interesting to speculate that perhaps we have an activation of a myeloid cell that's specifically being regulated by galectin-1. What I can show you in the time being is, again, if you take natural killer cells and combine them with Gowin-deficient glioma cells, in culture, you can get a certain amount or certain level of tumor-specific cell lysis. Well, what we can now show is that if you take a monocytic myeloid cell and add it to these two, you can effectively double the amount of tumor-specific cell lysis that you get in your cell culture. So this is a very early sort of result that we have. We have a lot more work to be doing to see if it's either direct or indirect. But some of the work that we've been showing right now is that it's perhaps a very complex interaction between the monocytic myeloid cell and the natural killer cell. There's some sort of crosstalk which still needs to be worked out completely. But suffice it to show that what we can find is that if you go back and you take recombinant galectin-1 and now you add it to your cell culture media, what we can show is that you dose-dependently decrease the amount of tumor-specific cell lysis that you see, all the way back down to what you would have if you only had natural killer cells in the well by themselves, which implicates very nicely a role for galectin-1 in the suppression of the pro-inflammatory phenotype that we're seeing in these myeloid cells. So it may be that galectin-1 is not influencing the natural killer cell directly like we thought, but in some way it's causing an anti-inflammatory immune response in myeloid cells, which are then critical to activate natural killer cell function. So in conclusion, what I've just shown you is that we believe that galectin-1 blocks tumor eradication through a tripartite mechanism, which involves entry, suppression, and resistance. So in mechanism 1, which involves entry, we can show that galectin-1 is somehow blocking the infiltration of GR1 positive myeloid cells into the tumor microenvironment. And as I showed you before, if you immunodeplete this population, you can get tumor outgrowth. So it implicates a very strong role, a pro-inflammatory role for these myeloid cells in the rejection of this tumor. Of those myeloid cells that do penetrate this tumor, we can show you in vitro evidence thus far that suggests that somehow through a complex interaction between these three cell types, that the galectin-1 that's expressed by these cancers is somehow causing an anti-inflammatory phenotype in those myeloid cells and downregulating their immune response. And lastly, mechanism 3 involves resistance. Again, one of the first slides I showed you suggested that somehow galectin-1 expressed by a cancer cell is a way to actually cause a resistance to inherent NK-mediated tumor lysis. So when you combine all three of those phenotypes, it explains, or at least helps to explain, why GAL1-expressing cancers or brain cancers suppress innate immune responses. And with that, I'd just like to thank my supervisors, Dr. Pedro Lowenstein and Dr. Maria Castro. I'd like to acknowledge many of the other members in my lab, and I'd like to specifically point out Dr. Vivek Yadav, a postdoctoral fellow in our lab, Peter Chalkley, a graduate student, and Rob Doherty, who are co-authors on the cancer research paper that I just showed you at the very beginning of the talk. And I'd like to further point out Daniel Zamler, who did the Kaplan-Meier survival analysis that I showed you. And with that, I'd like to take any questions. Yes, sir.
So, very nice work. How do you think you can harness this therapeutically? I mean, how are you going to deliver galectin-1 blockade to the CNS?
Yeah, so people have been developing small molecules as well as antibodies that specifically target galectin-1. But I feel like it seems like the mechanism is a lot more rich than that. And I think that instead of just blocking galectin-1 directly, perhaps what we could be doing is asking what is the effect, or how can we heighten the pro-inflammatory phenotype in the myeloid population. The cytokine arrays that I showed you indicated that there were potent myeloid chemoattractants that are upregulated in GAL1 deficient glioma. So from the therapeutic point of view, and I should say that galectin-1 is expressed in human cancers, human malignancy, and it actually correlates with the grade of those cancers. So if you have a lot of galectin-1 in that media, one would wonder if somehow you could artificially induce cytokine stimuli or sort of chemokines in that microenvironment to actually drive in pro-inflammatory monocytes, for example. So I think there's a lot of different ways that one could go with it. I tend not to think directly targeting galectin-1. It's going to be hard to target all of those cells all at once, but it remains to be seen.